Okay, today we're going to talk about germline editing in mice, which is to say, how you make transgenic mice. Okay, so there's traditional ways that have been around for a really long time. These mice are those whose genomes have been altered by DNA manipulations, and the oldest way, which is now back in fashion, is to inject the intended transgene into zygotes. Now, this was a technique which was extremely powerful, but it was not possible to get knockouts. But you'll see that the addition of CRISPR makes it possible now. The newer way uses ES cells, embryonic stem cells. It's not easy to get knockouts, but it is possible. But again, ES cells are still in use because some of the things that people want to do are a little more subtle, requiring the use of a little bit different technology. But we'll see. Okay, so the old way, which is back in fashion, is to inject zygotes with the intended transgene, or CRISPR components, or both. Okay, so what you have is a female mouse who's going to be treated with hormones in order to be superovulated. She's mated with a male mouse, and fertilized eggs are produced and collected. So those fertilized eggs are then injected with the intended transgene, or CRISPR components, as the case may be, and then cultivated. And then after a little bit of growth, they're transferred to a pseudo-pregnant female who has been treated with hormones to accept the newly fertilized eggs. And then she will then give birth to mice. And the little baby mice are then tested to see whether they actually acquired the transgene or not. So the donor mouse has to be superovulated. I think I said all this. Okay, so they're genotyped with the desired genetic events. Okay, so the old problem is that injecting a plasmid DNA doesn't guarantee much of anything. Even if the DNA actually binds itself into the genome, there's no control over where it goes, however many copies it goes. It certainly mostly does not go to its own homologous location. And when it does occur, you get many, many copies many times. And the baby mice are at best heterozygous for the transgene. So if you want to get homozygous mice, you have to then breed them to get homozygotes. But because of the fact that the DNA will integrate into the chromosome at basically random locations, it's virtually impossible to get knockouts in the standard method. Okay, CRISPR has changed all that because of its high efficiency and its ability to target specific genomic locations. So now you can use CRISPR to make transgenic mice by this method. You get your superovulated female. You then acquire single-cell zygotes and then you inject the single-cell zygotes with the CRISPR components. Those are cultured to the two-cell stage, reimplanted, and then founder mice are obtained. Now the difference here is that CRISPR is able to direct the transgene, or direct itself really, to the location in the genome where the desired changes can be made. So you don't have the problems of random integration, multiple copies, and unknown location. It's very different now. Okay, so you can inject the super-ovulated females, or the zygotes rather, and they will be injected with the synthetic guide RNAs, mRNAs encoding Cas9, or RNPs, including the Cas9 protein, etc. So these results are very efficient. So in the example I'm going to show you, you'll see the frequency with which the desired outcomes were obtained. Okay, so advantages of CRISPR is very efficient, very specific, although not totally so. And it will edit the target locus and not incorporate at random. Okay, it can be used to make knockouts. And importantly, it can very often edit both alleles in the diploid. So we often have baby mice that are homozygous for the editing event, even without any kind of breeding. This is huge. Okay, so here's a result of an experiment in which knockouts for the TET1, TET2, and TET3 genes were injected into the zygotes. Now, you don't have the best yield of baby mice necessarily, because, for example, with high levels of TET1 that the injected 35 zygotes, and only 19 were transferred, and of those they only got 2 newborns. But of the 2 newborns, 2 of them had both alleles knocked out. Similarly, for TET2, we had not the best yield of transferred embryos or newborns, but of the newborns that we got, most of them actually had both alleles knocked out. Similarly for TET3, although there's a little bit of a problem with TET3. So this demonstrates that this is a very efficient process and targets the correct locus in these embryos. You can even do 2 at a time. Here's an experiment where 2 attempts were made to knock out 2 genes, TET1 and TET2, and again they transferred quite a few embryos, 144 of them, of which only 31 gave actual newborns, but of those, a reasonable fraction, a high fraction really, had at least some mutant allele, and mostly both alleles were mutant in the embryos. So this is a total game changer as far as this type of technology is concerned. Okay, now embryonic stem cells are a very totally different technology, but even so, some of the subtleties of the desired mutations make them important, because the homology-directed repair that people want to do often is not very efficient and can yield off-target effects. So ES cells still have their place in this technology. Okay, so what ES cells are embryonic stem cells. These are maintained as cell lines, but were originally obtained from early embryos. So we're showing a picture of a very early embryo, a blastocyst, and it has the inner cell mass, and the cells within the inner cell mass are those that are going to produce the actual embryo. Okay, so those cells from the inner cell mass were cultured separately as a tissue culture line, and they're kept in the non-differentiated state. So what they can do is just divide in the proper culture conditions to produce more cells of the same type in non-differentiated state. Now, in that cell line state, they can be transfected, electroporated, or whatever, with whatever gene of interest or whatever manipulation of interest somebody wants to make. And so those are shown with the little red dots that you can select for the desired event and grow only those cells. And once you've got the desired cells, you can re-inject them into an embryo to produce transgenic mice. Okay, so for knockouts, this means that you can actually attempt to make the knockout in vitro using the cells, and then test the cell lines that you get to obtain the correct knockout. So look to make sure that it actually replaces the endogenous sequence. And so this means that you can screen through a lot of cell lines to find the one you want, but you don't have to make a mouse in the process until you've actually found the cell line that's producing the desired change. So once you identify the cells that have got the desired mutation, they can then be used to transfer to a blastocyst to grow up a baby mouse. So that's illustrated here. We're going to use a fine needle to take the desired ES cells that we've manipulated, inject them into a blastocyst, and then implant them into a foster mother. Now, the baby mice that come from this have 2 sets of parents, really, because the blastocyst that was injected already has a diploid genotype. And now we're injecting more cells with a different diploid genotype. So what arises is a chimeric mouse, which arises from those 2 types of cells, one type from the original embryo, and one type from the actual injected ES cells. Now, typically people use genetically mice with different fur colors in order to distinguish the chimeric mice and also to distinguish the progeny. So in order to get an actual transgenic mice, knowing that a lot of the cells are actually derived from the original embryo, what you have to have is the ES cell contributing to the germline so that when the mouse is bred, you will get animals that are actually derived from the ES cells. Okay, so I think I said this. So the idea is you put the ES cells into the blastocyst once you've chosen the ones you want, but they join with the other cells of the blastocyst and commingle with them to create the development of a mouse. And so it's helpful to have the ES cells to be of a different genotype as far as its fur, so you can see whether the baby mice are actually chimeric or not, meaning that they've got some cells from the ES cells and some cells from the original blastocyst. Okay, so here's some baby mice. These are obviously chimeric. They have patches of dark and light fur, reflecting the fact that they have 4 parents, 2 contributing to the diploid ES cell and 2 contributing to the original blastocyst. Okay, so this is represented here as a way of showing how this looks, that you have an embryo, which is albino, a blastocyst, and you've injected the ES cells, which are from a dark fur mouse. So now you take that chimeric mouse, made it 4 times the same albino mouse, and some of the progeny will be from the germline from the original blastocyst, but the others of the progeny will be from the ES cells. And so you can distinguish the progeny based on their fur color. And you can distinguish the successful transgenic animals based on the different patches of color throughout the body of the animal. Now, the chimeric mice must be bred to obtain non-chimeric animals. So it's obviously crucial that the ES cells contribute to the germline. And then you usually need to breed them again to get homologous lines of mice. All right. So in terms of doing this sort of stuff, this isn't just a chip-seek experiment. Not everybody can do this. Not every lab can do this, really. So there are companies that specialize in making these for people who want them. And there are also huge collections of mice that already have knockouts available that you can purchase or get to contribute to your own research. Now, one of these is the International Mouse Phenotyping Consortium. And this is a huge consortium involving more than 20 labs internationally to make and characterize these various knockout mice. Okay, so the goal is to build a comprehensive catalog phenotyping knockout mice of ideally of all different genes eventually. And already there are 8,500 different genes knockouts that have been made and phenotyped. All the data are available, and the mutant strains are also available. And they've done a lot of stuff already to model various human genetic diseases, and of course they found that quite a few genes have embryo lethality defects, and those are ones we need to worry about later. Okay, so this consortium is still very, very active, and they're continuing to develop more mouse strains, more mouse collections using improved technologies, and also, importantly, to create mouse models of human genetic diseases, and many, many more projects. Okay, one thing to think about is that you can't just knock out any gene, because some genes are essential for life, right? If they're lethal, if they're knocked out, you can't make a mouse line with that knockout. And clearly, many of the embryonic lethal genes are those that are associated with disease. So naturally, we need to have conditional alleles, or at least heterozygous alleles, in order that we can study what's going on with those particular genes. So I need to remind you about homologous recombination to understand this. This is something we've seen before. And remember, if you make first step in making CRISPR is to make a double-strand break at the desired location using Cas9, etc. And the two outcomes are either non-homologous end joining, or if you have a homology repair template to basically replace the sequence that is there with your sequences within your homology-dependent repair pathway. Okay, we've seen this before. Here's what happens with non-homologous end joining. You make a double-strand break. Mutations arise because processing of that double-strand break in order to put the two pieces of DNA back together again usually leads to the deletion and sometimes insertions of a small number of nucleotides. Okay, so you can do that using CRISPR. For example, if you just do a double-strand break at a crucial exon, even if you don't have a homology repair template, you can make a frame shift easily because non-homologous end joining will delete a few nucleotides. Also, you can make a double cut within the exon, and when non-homologous end joining puts those together, you'll end up with a deletion. Example. Right, so the examples are what I just showed. Now, if you want to use homology repetitive repair, that works too, but I'm just going to look at this third example, number 3. You make your cut at a critical exon, and you can provide the cell with oligonucleotides that contain the desired replacement. Instead of just deleting nucleotides, you're going to substitute something else. And this example is for a point mutation that you'd like to study. So manipulations like that are really quite frequent. Okay, so this attempts to make a point mutation in a particular gene with the resulting repair that will, at least some of the time, contain the desired, well, replace what was there with the desired point mutation, right? But sometimes you get non-homologous end joining anyway, and sometimes you get repair. So you can never really guarantee that until you've actually looked and see what you get at the end. Okay, so another version of this is what a knockout would look like, where what you're trying to do is actually delete the entire gene, in which case you're going to have recombination between the gene itself and the incoming plasmid, which is going to be homologous on the ends to the flanking region, and recombination will replace what's there in the gene with what you've put in for your homology-dependent repair pathway template. So conditional alleles are more subtle than that. They use bacterial or yeast recombinases to delete specific exons. Okay, so we're going to have to back up a little to look at what these recombinases are about and the consequences of recombination between inverted and direct repeats. All right, so the CRISPR system from bacteriophage P1 is the most commonly used, but there's another one called the flip system from Saccharomyces. Okay, they're very similar in their approaches. Okay, so these are both site-specific recombinases, which bind to specific target DNA sequences and recombine them. So when the two sequences are in direct orientation, recombination between them will delete the region between them, reminding you of the allele recombination, which we've seen. So that we can set up this system using recombinase target sequences, which are called lock sites, flanking the exon to be deleted. So these are called Floxed alleles. Okay, so this is a little complicated. All right, so the Crea recombinase knows how to do a recombination event between two lock sites. The lock sites are 34 nucleotides long, base pairs long, and what we're going to do is set up a lock site on either side of the exon that we want to delete. Now, if Crea is then introduced to that exon, what it will do is do a site-specific recombination between those two lock sites, and the result is deletion of the exon in question. Okay, now we need to provide Cre, and in most systems, the Cre is provided by a different, a construct where a promoter is driving Cre, but usefully only in certain cell types. Okay, so a reminder about recombination between direct repeats. It leads to deletion of the intervening sequence. We've seen this slide before. So what happens is if you're going to recombine these, you have to kind of contort the DNA around such that the direct repeats are aligned in the correct direction with each other. And then the resulting recombination will lead to a deletion of the region between the two repeats. Okay, so here's a different example involving a virus. You've seen this slide before also, that you simply line up the direct repeated sequences in such a way that they're pointing in the same direction, and then allow the recombination to occur between the two. And the result is deletion of this region, including one copy of the repeat. So what that looks like, you will have a gene, you do the recombination, and the next gene. And so what you end up with is a chromosome really, that is missing that section between. And then the other recombination product is a circle which contains one copy of the repeat and the DNA that it was originally between the two direct repeats. So here's what this looks like as far as the deleted region. In purple, we have the deleted region, and now it's gone, and the outcome is that you've deleted the region that was originally, in this case a virus, that was originally between the two direct repeats. All right, so we're going to use the CRISPR system to make a conditional allele. And to do that, we have to make a Floxed gene, Floxed allele, which is say we're going to create an exon which has a LOX P site on either side of it. So those two LOX P sites, one on either side in the direct repeat orientation, will be sitting there in the chromosome. Now this will not cause any harm since it's sitting in the adjacent intron, but as long as there's no recombinase around, it's going to pretty much stay put. But if we put the Cre protein in, recombination will occur, and the exon will be deleted, and the gene will be inactivated. So this is, in a sense, a conditional allele, in that the gene is active. The Floxed allele is harmless, usually, until the Cre recombinase is provided, and that makes the deletion. Right, so back to our system here. We have the Cre recombinase, and we have the action on a piece of DNA, and then we have the promoter that's driving the Cre. Right, so what this looks like, if we put in a Floxed allele on a plasmid, here's our Floxed allele. The little yellow things represent the LOX P sites, and they are sitting on one on either end of the exon. If we hit both ends of the exon with CRISPR, that will promote recombination between the LOX sites and the endogenous allele on the chromosome, leading to replacement of the endogenous allele with now the Floxed allele. So that's the Floxed allele, which is potentially susceptible to the Cre recombinase. But there's no Cre recombinase around right now in order to cause the recombination, so it sits there pretty harmless until Cre is introduced. But if we introduce Cre, recombination happens between the two LOX P sites, and the exon is deleted, leaving only one LOX P site in its wake, but deleting the entire exon. So that, if that's a critical exon, that would be an inactivating mutation in that gene. All right, so the first thing is you make a Floxed allele of a gene in a mouse. And now what? Well, now you have to breed the mouse to a Cre mouse that expresses the Cre in the right cells. Okay, and that is key here. We want to have the Cre mouse basically express it only in certain cells. You don't want to knock it out in the embryo. You want to inactivate it in the liver, eye, intestine, brain, or whatever particular tissue you want to see the effect of deletion of that exon. Right? So if it's a liver-specific protein, for example, you want to express the Cre in liver. For that purpose, you need a promoter which is only active in liver. So this is accomplished by crossing your mouse to a mouse that we have a liver Cre already, that these mice have been set up to deliver Cre to the intended tissue of interest. Okay, so what we're representing here is a mouse that has a normal copy of the gene of interest, but it's got a liver-specific Cre in that the Cre is being delivered by a liver-specific promoter. Then when we breed that mouse to a Floxed mouse that's got that particular allele on both chromosomes, actually, if we breed those, what happens in the heterozygote is that the Cre will be activated in the liver, and it will delete that exon in the liver. Right? Still, we're only heterozygous, and if we want to make a homozygous, we need to breed it some more. But the idea is you don't want Cre to be active in all of your cells, and so you need to have a promoter or a series of promoters or a series of mice which express Cre in different tissues in order to selectively knock out your exon of interest only in those cells. Okay, so we'll mate the mouse to a Cre mouse, and that will give us mice that will remove the Floxed allele. only in the cells we're interested in. And then we can infer the function of the Floxed alleles in those cells. So diagrammatically, in these particular mice, we started with our Floxed allele, we introduced tree, we put them together, and the gene of interest was knocked out only in the target tissue. There are many, many sources of Cree mice, and people buy the Cree mice that will produce Cree in different tissues, etc., as needed. So it's actually more complicated than that, but we're going to leave this alone. We're not going to worry about this very last slide. I hope you got the idea, though.